The study of genetics stems from the dedicated work of the monk Gregor Mendel and his study of peas, and later from pioneers such as Thomas Hunt Morgan, who worked with the Drosophila fruit fly. Choose a lesson and then press click here to begin.
Dominance.
In this lesson you will do the following. Learn about Mendelian inheritance. Follow and participate in Mendel's pea lab experiment. Examine patterns in generations of peas. Observe the law of dominance of alleles.
Welcome to the Mendel Pea Lab. In this lab we will show you why the Augustinian monk Gregor Mendel is considered the father of modern genetics. We will also show you how he used the scientific method steps of observation, hypothesis, and experimentation to form three very important conclusions about heredity. Heredity is how offspring biologically inherit physical characteristics from each of their parents. And genetics is the study of heredity and the mechanics behind this process. The portion of genetics we will be covering is formally called Mendelian inheritance. Perhaps you've wondered, as our ancestors did, why we ourselves look so much like our parents. Were these traits blended from our parents' traits? Or were they shuffled together like a deck of cards? The monk Gregor Mendel wanted to know how heredity worked, so he set up experiments to observe traits passed down from successive generations of pea plants. In 1865 he published these results in a paper entitled Experiments in Plant Hybridization. During his lifetime this paper attracted little attention from scientists. It was not until the beginning of the 20th century that his work was combined with that of other early geneticists. Let's now help Mendel in his earlier work by following along in his early hybridization experiments.
The purpose of these experiments is to observe the patterns by which differing plant strains combine into hybrid forms, and then try to deduce a mechanism that explains why these results occurred. This is a green pea. It is of the family Leguminosae and species Pisum. This is a yellow pea. It is of the same species, but it has a different colored trait in the part you eat called the albumen. Both of these peas came from parents that were the same color strain as themselves, and given the chance to self-pollinate, as adults the green pea will produce only green peas, and the yellow pea will produce only yellow peas. I call these peas true-bearing, but these days they are more often called homozygous. There are 6 other traits I found that I am simultaneously testing, along with this experiment. There are a total of 7 traits, which are as follows. 1. Seed color, yellow or green. 2. Seed texture, round or wrinkled. 3. Pod appearance, inflated or constricted. 4. Pod color, green or yellow. 5. Flower position, axial or terminal. 6. Plant stem length, long or short. And lastly was 7. Seed cover color, white or grayish-brown, which incidentally coincided with flower color white, or purple. Each of these traits has 2 contrasting strains of homozygous pea plants. It has been known for a long time that crossing 2 homozygous strains should get a hybrid pea, or as geneticists now say a heterozygous pea, but until now no one has observed this with the scientific method. Through careful observation, I would like to see if any patterns in offspring type occur. If they exist, then it is my intent to decipher them with an explainable law. So let's begin first by planting our homozygous parent peas. Good. Pea plants usually take a couple of months to mature. We shall call this generation of plants the parental P1 generation. Now we are going to cross-pollinate both plants. And it can be physically done 2 ways. We can either take the pollen from plant P1 green and place it in the flowers of P1 yellow, or we can do the reverse and move the pollen from P1 yellow and place it in the flowers of P1 green. Because we want to determine if there is a difference between the P1 green and P1 yellow offspring, we need to do both crosses. To begin this procedure, we need to choose a donor flower on each parent that we shall get our pollen from. All other flowers must have their stamens removed before they fully mature. Using the forceps, remove the underlying hood on each of the flowers, and also remove the stamens in the P1 flowers that we will pollinate. This is to prevent these flowers from self-pollinating with their own stamens. To do the P1 cross, we need to take a clean brush and gather pollen from the P1 green donor flower, and brush the P1 yellow flowers with it. Very good. Now with a new clean brush we need to do the reverse P1 cross pollination also. Good job. Now snip off the self-pollinated donor flowers. Any pollen that lands on a stigma will be transported through the stylus to the ovary. When the sperm from the pollen contacts an egg cell there, a pea embryo is formed. The number of successful fertilizations will be equal to the number of peas. Now that the pea pods are completely ripe, pick them off. What physical traits, or phenotypes do you think are in the pods? A. They are green peas. B. They are all yellow peas. C. They're a mixture of yellow and green peas. D. There is a new color of pea. To find out, we need to open the pods and observe the first filial generation F1. As you can see, all the peas are yellow, regardless of the direction of the P1 cross. It therefore seems that the yellow factor is dominant, and the green factor is recessive for any crossed first filial F1. Now, let's see how the other pea plant traits I crossed came out. For seed texture, all the peas were round and none were wrinkled. For pod appearance, all the pods were inflated and none were constricted. For pod color, all the pods were green and none were yellow. For flower position, all the flowers were axial positioned and none were terminally positioned. For plant stem length, all the plants were long instead of short. And lastly, for seed cover color, all the seed covers and flowers were white, and there were no gray seed covers or purple flowers. In every crossbred case, one of the strains of plants dominates the trait of the offspring, and somehow masks the behavior of the other. Surprisingly, dominance occurs independently from whichever plant bears the seeds or donates pollen. This gives us a clue that whatever factors are taking place, sperm and egg cells in each plant seem to have factors equal in magnitude towards the outcome of the process. The fact that one phenotype completely masks the physical traits of another, is what I shall call the principle of dominance. What became of the recessive trait? Did it vanish? Or will it show up again in the second filial generation called F2? We shall find out in our next lesson.
Law of segregation.
In this lesson you will do the following. Plant an F2 second generation of green crossed with yellow peas. Determine patterns if any in offspring. Use a Punnett square to validate our observations. See how the concept of alleles validates our observations. In our last lesson, we saw there were no green peas in the first generation F1 cross between true-bearing green and yellow peas, and only the dominant yellow-colored peas grew. This planting season we will do something different, and self-pollinate each F1 pea plant. This is to see if these peas are true-bearing like their parents. So let's begin by planting a first-generation F1 pea, and letting it grow. In order for this plant to self-pollinate, we shall simply leave it alone. Once we allow these F1 peas to germinate and grow, we can then remove the pea pods. Good. Now let's open them up to see what's inside. Very interesting indeed. There are now green peas showing up in the F2 generation, which means the F1 peas were definitely not true-bearing, nor homozygous. So now, approximately what percentage of the F2 peas appear to be green? Correct. This is actually a small sample of my original experiment, where I actually planted all the F1 peas, and counted a total of 6,022 yellow peas, and 2,001 green peas in the F2 generation. From the results, there seems to be a 3-to-1 ratio of yellow to green F2 peas. This means that the green factor must have been hidden and stored by the dominant outward appearance or phenotype of the F1 yellow peas. If this is so, we can hypothesize that there are two factors coexisting in each pea, which we shall call alleles, one randomly given from each parent's pair. To standardize a way of representing this, let us name our P alleles with a capital Y for the dominant yellow allele, and a lowercase y for the recessive green allele. Here we have each of our original parents. Note that the yellow P has two big Ys, and the green P has two small Ys. These represent the two alleles each has. If we cross P1 and P2, each parent will randomly give one of its two alleles to the F1 offspring in the form of pollen or an egg cell. This is most easily represented with the tool called the Punnett square, created in the early 20th century by R.C. Punnett. By placing the alleles of each parent along the horizontal and vertical axis, we can build a table that organizes each type of allele pair. We can then add up the types to determine the probability of genotype. Since big Y represents the dominant yellow color, any P that has at least one big Y will have the dominant yellow phenotype. For the case of the F1 hybrids, which all have one big Y and one small y, there is a 100% probability of yellow-looking hybrids, or phenotype. This matches and supports our original observations in the dominance lesson perfectly. Alright then, how will the Punnett square predict the growth of the F2 generation that we already observed? If we select any one of these yellow F1 peas, grow and self-pollinate it, we must treat it as a cross with itself. Fill in the F2 allele pairs of the Punnett square. Great job! Based on these results, we can see that 25% of the predicted Ps are homozygous dominant yellow, 50% are heterozygous yellow hybrids, and 25% are homozygous recessive green. That's a 3-to-1 ratio of yellow to green, just like we saw in the beginning of this lesson. So our phenotypes from our predicted and actual Ps do match. How then can we validate the genotypes of our Punnett squares result? Since our prediction said that 25% of our F2Ps would be homozygous yellow, we need to cultivate a self-pollinated F3 generation to see if this is true. Press on each real F2P to plant it, and see its F3 offspring. Excellent! We can see that 25% of our F2 plants were indeed homozygous dominant yellow, 50% were heterozygous yellow, and 25% were homozygous recessive green. In actuality, I repeated these experiments many times over, and my hypothesis would eventually become known as Mendel's first law, or law of segregation, which by definition states 1. For every physical characteristic, an organism carries two factors. Technically, these would one day be called genes, and variations such as color in these would later be called alleles. 2. Each parent segregates its pair of factors during gamete sex cell production, and randomly contributes one of these alleles to its offspring. Over the last century and a half, the reliability in this law has been rigorously tested with many organisms, and it still holds true as a basic underlying premise of genetics. You have now completed this lesson.